Seed-bearing plants face a strict resource problem. They must guarantee the heavy metabolic cost of packing a seed is paid only if a viable embryo forms. Angiosperms solved this through a highly synchronized dual biological mechanism known as double fertilization. This diagram shows the setting for this mechanism, the embryo sac. Before fertilization, this microscopic structure is organized into 7 distinct cells holding 8 total nuclei. 2 specific targets matter most: a single haploid egg cell at one end and a large central cell containing 2 polar nuclei in the middle. When a pollen tube reaches the ovule, it breaches the sac, delivering 2 haploid male gametes. This dual delivery triggers 2 simultaneous fusions. Both must succeed perfectly for the resulting seed to be viable. The first event is syngamy. 1 haploid male gamete fuses directly with the haploid egg cell. This yields a diploid zygote, restoring the paired chromosomal state. Simultaneously, the second male gamete migrates to the central cell and fuses with the 2 polar nuclei. Because 3 haploid nuclei merge, this triple fusion creates a triploid structure, the primary endosperm nucleus, or PEN. The surrounding central cell housing this new nucleus is officially renamed the primary endosperm cell. These 2 new bodies have entirely different functions. The zygote will undergo embryogenesis to become the actual plant. The primary endosperm nucleus will rapidly multiply to build the endosperm, a massive nutrient-dense storage architecture. To ensure a food supply is ready when needed, endosperm development typically begins before the embryo even starts to grow. The embryo cannot survive alone. Its future depends on the exact mechanical pathway the primary endosperm nucleus takes to construct this triploid nutrient factory. The physical architecture of the endosperm is determined by a single mechanical variable, the precise timing of cell wall formation relative to nuclear division. Biologically, these 2 processes are karyokinesis, the division of the nucleus, and cytokinesis, the formation of the cell wall. The most common developmental pathway is nuclear endosperm. Here, the primary endosperm nucleus undergoes rapid and repeated karyokinesis. The defining feature of this pathway is that these successive nuclear divisions occur without any immediate cell wall formation. As the cell expands, a large central vacuole forms. This physical void pushes the thousands of free-floating nuclei outward to the extreme periphery of the cell. This pathway is dominant in major cereal crops like maize, wheat, and rice. By delaying cytokinesis, the plant rapidly generates a massive, liquid, multinucleate pool of nutrients, prioritizing volume expansion before committing to structural rigidity. The second developmental pathway is cellular endosperm. The mechanics here are the exact opposite. Every karyokinesis is instantly followed by cytokinesis. Because a wall forms after every single division, this endosperm tissue is strictly multicellular and structurally solid from the very first split. Plants like datura, petunia, and balsam utilize this cellular pathway. To see both pathways operating in a single organism, take the coconut. The liquid coconut water in the exact center is free nuclear endosperm, a fluid suspension of thousands of free nuclei. The surrounding solid white kernel, or meat, is cellular endosperm. In coconuts, cytokinesis begins at the periphery and slowly works its way inward, leaving the center locked in a free nuclear liquid state. The coconut demonstrates how the same triploid genetic material builds entirely different physical states, based purely on localized timing of cell wall formation. The third pathway is Helobial endosperm, an intermediate type predominantly found in monocotyledons. It begins precisely like the cellular type. The very first nuclear division is followed by an immediate cell wall, but this first division is intentionally unequal. It partitions the sac into a very large micropilar cell and a much smaller chalazal cell. From there, the process shifts. The large micropilar chamber switches entirely to the nuclear pathway, undergoing rapid free nuclear division, while the small chalazal chamber remains largely static. The classic example of this intermediate pathway is Arameris. Helobial endosperm acts as a structural hybrid. It utilizes an initial cell wall to physically partition the seed, then uses rapid liquid nuclear division within that partition to optimize nutrient scaling. Once the endosperm architecture is built, its final form depends on how the embryo consumes it during maturation. The endosperm is a transient tissue. Its sole biological purpose is to provide starch, proteins, and lipids to the growing embryo. This schematic cross-section of a mature wheat seed illustrates the first outcome, albuminous seeds. In an albuminous or endospermic seed, the embryo does not completely consume the nutrient layer during early development, so the endosperm persists into maturity. On the right side, we see the second outcome, an exalbuminous seed, where the endosperm is entirely absent. Here, the growing embryo completely digests the endosperm before the seed matures. The plant transfers and stores those nutrients directly into the embryo's own thick cotyledons. Wheat, maize, castor, and coconut all retain their endosperm in the final mature seed. Peas, beans, and groundnuts follow this exalbuminous strategy. Whether a seed is albuminous or exalbuminous dictates exactly where its energy reserves are physically located when it enters dormancy. The logic of angiosperm reproduction is strictly sequential. Double fertilization guarantees that a primary endosperm nucleus is only formed if a fertilized zygote already exists. The timing of karyokinesis and cytokinesis then engineers that nucleus into nuclear, cellular, or helobial endosperm. Finally, that tissue is either preserved for germination or cannibalized early by the cotyledons. The evolutionary dominance of flowering plants is built upon this sequence. They conquered terrestrial ecosystems by engineering a mathematically precise, on-demand triploid nutrient factory.